And the final talk in this session of Understanding Why ALS Occurs, it is my pleasure to present a postdoctoral fellow at the Université Laval, working with Dr. Yves de Koninck, and prior to that, working with Dr. Melanie Wooden at the University of Toronto, is Dr. Sahara Khademullah. She's going to talk about inhibitory homeostasis as an unlikely suspect in ALS. So welcome, Sahara, and thank you so much for joining us.
Hi.
Hi, we can hear you and see your presentation.
Okay,
so,
great. Great.
Thanks.
That's great. Thank you. So, as my title suggests, I'm going to be talking about the role that inhibition plays in the onset and progression of ALS in the SOD1 mouse. And I'll focus mostly on the data that I collected in my PhD with Dr. Melanie Wooden at the University of Toronto. And if we have some time, I'd like to finish up by telling you a little bit about what I'm working on in my postdoc currently with Dr. Yves de Koninck. But first, of course, I'd like to say thank you to ALS Canada for inviting me to speak at this forum. And I'd like to start my talk today by telling you first why it is that I've been interested over the past few years in understanding the role of inhibition in ALS. So as many of you are aware, in the central nervous system, it's largely governed by two key transmitters: glutamate, primarily considered an excitatory neurotransmitter, and GABA, an inhibitory transmitter. And a balance between excitation and inhibition in the CNS is essential for normal brain function. So if this balance is disrupted, it can lead to neuronal hyperexcitability and the onset of various neurological disorders. And to date, we know that it can lead to neuropathic pain, schizophrenia, and depression, among other disorders. So of course, one outstanding question is exactly how this equilibrium is maintained and what triggers its disarray. So it's been well documented in the literature that hyperexcitability is in fact a hallmark feature of the pre-symptomatic phase of ALS. However, much less is known exactly about how inhibition is affected during this phase. So the main goal of my research over the past few years has primarily been to determine if the reduction in the strength of inhibitory synaptic transmission and what is its actual role in the pathogenesis of ALS. Sorry, there you go. So as I mentioned previously, there is strong evidence implicating cortical hyperexcitability as well as the dysfunction in the primary motor cortex of ALS patients and rodent models of ALS alike. So this, of course, is important as it's this brain region, shown up here, that contains the motor neurons that then extend down into the spinal cord down here, that then go on to produce voluntary movements. Furthermore, these excitatory motor neurons are pyramidal cells located here in layer 5 of the primary motor cortex, or the M1, receive direct inhibitory inputs from a variety of inhibitory interneurons, as can be seen here in blue, green, and yellow. Of these inhibitory interneurons, parvalbumin-expressing interneurons account for approximately 50 to 60% of all interneurons here in layer 5, and are the predominant source of inhibition as they synapse directly onto the cell body of the pyramidal cell. While the second largest population account for approximately 20 to 40% of interneurons, and these are called the somatostatin interneurons, these now project onto the distal dendrites of these excitatory pyramidal cells. Thus, with the primary motor cortex containing such an intricate relationship between both inhibitory interneurons and excitatory pyramidal cells, it was our thinking during my PhD that the presence of cortical hyperexcitability in ALS might in fact be the result of an imbalance between excitation and inhibition. So a lot of our reasoning for this line of thought was in part found here amongst these recent papers. So a research group led by Steve Vucic from the University of Sydney, which focuses on identifying and understanding changes in cortical activity associated with ALS, recently identified the presence of cortical hyperexcitability in a subset of ALS patients carrying the SOD1 gene mutation. So of course, as many of you are aware, the SOD1 gene accounts for approximately 20% of all inherited or familial forms of ALS, which also make up about 10% of all ALS diagnoses. An additional 3% of sporadic forms, which then make up the remaining 90% of cases. So while this finding is interesting, even more significant was Vucic's finding that cortical inhibition was also reduced in these patients, and this appears to precede the onset of cortical hyperexcitability. Additionally, a more recent paper shown here from Zhang and colleagues from the NIH found that somatostatin interneurons were hyperactive in the TDP-43 mouse model of ALS, which can be seen here, while the PV interneurons were hypoactive, as seen here when compared to the control recording. So to be clear, you can see that there's far less spikes or action potentials when compared to the control recording. Together, these findings led us to ask whether there was a reduction in the strength of inhibitory synaptic transmission present in the SOD1 G93 mouse, and whether the chronic activation of inhibition in these interneurons, more specifically PV interneurons in the motor cortex, could actually delay the onset of the disease. So as mentioned, I use the SOD1 G93A mouse model for most of my experiments, as it is, in fact, the most widely used ALS model. In addition, this mouse reliably shows a behavioral phenotype, and most importantly, it has the early onset cortical hyperexcitability that we were really interested in studying. So first, to determine whether PV interneurons were in fact hypoactive in the SOD1 model, I began to crossbreed my SOD1 mice with PV-CRE mice as a means to identify these PV-expressing interneurons that I was interested in. So I used chemogenetics to target and activate these PV interneurons. And more specifically, as described here, I used the DREADDS, or designer receptive exclusively activated by designer drugs, which includes a group of engineered G protein coupled receptors, which are activated by inert ligands, and an example of which is clozapine N-oxide, or CNO. So with that, I virally delivered the chemogenetic activator, as shown here, HM3D, tagged with mCherry for visualization, to the PV interneurons in the L5M1, or the layer 5 of the motor cortex. And this receptor was then activated by its inert ligand, which is CNO, or clozapine N-oxide. As you can see over here in this diagram, or this figure, we managed to achieve approximately an 85% transduction rate of the PV interneurons with this strategy. So moving on, I then recorded from these PV interneurons in layer 5 of the M1 and found that they were in fact hypoactive. So these are the SOD1 recordings, and these are the wild type recordings. And hopefully you can see that there's essentially no spiking, and it's extremely low in the overall average. Additionally, I activated the G2 receptor that I injected with CNO, and I was able to see an increased firing when in the SOD1 mutation when compared to the previous without the CNO. So to determine if this increase in PV-mediated inhibition could affect the pyramidal cell firing, I recorded from the pyramidal cells of the layer 5 of the motor cortex and found that they were in fact hyperexcitable as we expected, and that the application of CNO, as seen down here in gray, was actually able to reduce this hyperexcitability in the SOD1 mouse back to that of the baseline in the wild type mouse. So now that I knew that we could essentially decrease this hyperexcitability in the layer 5 of the primary, or layer 5 of the motor cortex in the pyramidal cells with this strategy, I then wanted to test really were we able to delay the onset of the behavioral deficits associated with ALS in the SOD1 mouse model. So what I'll do is start off by describing this experimental paradigm in which at week 6, we surgically injected the HM3D receptor into the layer 5 of the motor cortex in the SOD1 mice and managed to target these PV interneurons as I had shown previously. We then increased their activity by pre-symptomatic administration of CNO, and this was provided to the mice via their drinking water, and they were freely able to drink as much as they wished. Between week 12 and 17, when the symptoms are quite visible in the SOD1 mouse, we began to take behavioral testing. And what I'd like to show you or draw your attention to first, before I begin to describe the behavioral graph seen in the lower panel, is that the red or the red dots and the red dotted line is the SOD1 mouse without treatment, essentially, while the blue line is the SOD1 mouse with treatment. And hopefully what you can see across all behavioral tests well into the symptomatic phase, so at week 17 of these SOD1 mice, is that across grip strength, hanging wire, and rotor rod, we had a significant improved performance in the SOD1 mice with treatment. And now what I'd like to show you here is another test that we performed, which is called paw print analysis. And hopefully what you can see is that the SOD1 mice without treatment took a significantly longer time to finish this test. In addition, their gait distance, both forelimb and hind limb, was much shorter. And a nice example of this is provided here in this panel. At week 17, so what this test is essentially, we dip the forelimbs and the hind limbs in different colored paints, and we allow the mouse to walk across. And we assess their gait as well as their distance that they can walk. And over here, you can see the wild type has a nice overplacement of both hind and forelimbs, and the paw prints are very, very clean. And over here, you can see the SOD1 mouse with treatment and SOD1 mouse without treatment. And without treatment at week 17, you see significant hind limb dragging and hind limb weakness as indicated by the smears or the paint smears, but you don't see this quite significantly in the SOD1 mouse with treatment. So we also wanted to now look up into the brain and see were we able to preserve any of the neurons or different cell types in the motor cortex. We used SMI32 labeling to label the motor neurons and NUN to label overall motor or overall neuronal health. And what I'd like to draw your attention to is the red panel over here, which are the SOD1 mice without treatment, and the blue here, which are the SOD1 mice with treatment. And over here, the graphs with the same color. So hopefully what you can see is that we were able to preserve both the number of motor neurons in the mice or the SOD1 mice with treatment, as well as neuronal health in these mice. We saw significant or similar results when we looked for PV interneurons. So if you remember, these are the interneurons that we actually targeted. And once again, with the mice with treatment, we saw a significant preservation in these interneurons when compared to the SOD1 mice without treatment. So while we found these results to be fairly interesting in and of themselves, we did want to find a way to make them more clinically translatable, since of course humans don't have CRE-recombinase and it's a little bit hard to target PV interneurons that way. So with that, we were lucky enough to find a publication from Gord Fischel's group which outlined their recent development of a virus containing a DLX enhancer. So they showed that this could specifically target GABAergic forebrain interneurons without the need of any CRE-recombinase across a variety of animals. So our thinking was that since gene delivery therapy is already being used in the EU and under several clinical trials in North America, we thought that including this virus in our treatment strategy would actually increase the translatable possibilities of our work moving forward. So we used the exact same experimental paradigm as we had with the PV CRE-SOD mice. However, at week 6, instead of injecting the HM3 virus that would only affect the CRE-recombinase or the mice with the CRE-recombinase, we injected it with the DLX enhancer to affect the GABAergic forebrain interneurons. And when we performed the grip strength, hanging wire, and rotor rod tests, what we saw were similar results to our previous strategy, in which the SOD1 mice without treatment began to decrease in their motor performance, while the SOD1 mice with treatment, as you can see with the blue line, significantly improved, or not improved, but delayed their loss of motor function. We then again went back to the motor cortex to assess our motor neuron loss and neuronal health loss. And we once again found that over here in the SOD1 mice with treatment, we were actually able to once again preserve the motor neuron population when compared to the wild or the SOD1 without treatment, and the neuronal health population when compared to the SOD1 mouse once again without treatment. And again, those PV interneurons that we were targeting were also preserved. So one important thing that we hadn't considered thus far, but then it came to our attention, was that ALS patients, of course, are usually diagnosed a year or two following the onset of their initial symptoms. And this is, of course, a very important issue to consider for our treatment strategy, with whether or not our treatment strategy could in fact be effective during the symptomatic phase of the disease. So to determine this, we then began to repeat our exact experimental design, which was over here. However, instead of giving the CNO pre-symptomatically and ending it at week 13, we began to give the CNO administration starting at week 14, so at the very early onset of the symptomatic period, and we did not stop it, but we continued it until the animal's natural endpoint. And what we found once again was that across grip strength, hanging wire, and rotor rod, that our treatment significantly improved all motor functions in the SOD1 mice with treatment. So again, you can see that in the blue line compared to the SOD1 mice without treatment in the red line. So to our surprise, of course, which is not often seen, we also found that the continuous chronic activation of PV interneurons also resulted in a decrease in the weight loss in the SOD1 mice, which we hadn't in fact seen in our previous pre-symptomatic intervention, where we only gave the CNO through the pre-symptomatic phase. So basically what we're finding here is that during the symptomatic intervention, when we chronically administered CNO and did not stop the treatment, we were able to see an increase or the preservation of weight in these SOD1 mice. And more importantly, we were able to actually see an increase in survival by more than a month in the SOD1 mice with treatment when compared to the SOD1 mice without. So in conclusion, we found that changes in the PV-mediated inhibition can be detected prior to the onset of symptoms in the SOD1 mouse. And this, of course, is in line with our previous findings, or not our previous findings, but the literature's findings, that there's a decrease in short intercortical inhibition in the motor cortex of ALS patients. In addition, increasing PV-mediated inhibition via DREADDS early on in the symptomatic phase can actually prevent cell death in the motor cortex and preserve both interneuron as well as motor neurons alike. So this strategy can also delay the onset of motor deficits and prevent the onset of weight loss in mice, as well as extend their lifespan by more than a month. So with this, we're now very excited to complete more preclinical work to determine ways to improve our current treatment strategy. And in addition, since I've moved now to the DeConic lab, my most recent research interest has primarily been focused on looking at different ways to really target and activate the inhibitory pathway via increasing the expression and function of various proteins that govern inhibition in both the brain and the spinal cord. And while much of my work to date has been completed in the motor cortex of ALS mice, I do recognize that motor neurons in the spinal cord are also not only very vulnerable in this disease, but also extremely important in understanding the progression of the disease. Thus, I'm very interested in moving forward to take a closer look at exactly how the entire cortical motor pathway from the brain and the motor cortex down to the spinal cord and in the reverse undergoes motor neuron loss, as well as changes in excitation and inhibition balance. So I'd like to end my presentation by thanking my former PhD supervisor, Dr. Melanie Wooden, and the Wooden Lab members, as well as some of our collaborators that have provided excellent advice and support throughout the process, which include Janice Robertson and her team, as well as members of the ALS team at Sunnybrook Hospital. And finally, I'd like to thank my current supervisor, Dr. Yves DeConic, and my current lab members, as well as the patients and their families, ALS Canada, Brain Canada, and the FRQS from Quebec. And I'd be happy to take some questions. Thank you.
Thank you very much, Dr. Khademullah. That was amazing. And actually, the results in the mice are pretty significant compared to some of the other things that have gone translationally. So
Right.
It really looks pretty exciting for a translational aspect. And if one quick question on that
Sure.
note, has there been any precedent set, and this could be a naive question, but any precedent set in terms of someone doing motor cortex inhibition or a reduction of excitability perhaps in a human, of any kind of human trial for any type of indication?
No, very little actually has been done in the ways of understanding how inhibition is altered in patients. The research that I showed by Steve Vucic is one of the very few things that have been done in patients to actually see that inhibition is actually altered in ALS patients. And this has recently been shown to be the case in not only SOD1 patients, but also patients that have the C9orf mutation as well. As for treatments that are currently being offered to patients, to the best of my knowledge, nothing has really been done to target inhibition, although it is thought that riluzole can affect both excitability of neurons as well as inhibition, but it's not very clear exactly how it's happening.
So when it does eventually move to human clinical trials, I suppose there will have to be a safety phase first to make sure that it's okay in humans to do this type of inhibition
Absolutely, absolutely. We want to, that would definitely be the first phase, because this is obviously very new, and not yet, nothing like it, I think, has been done in patients. So we would first probably try a few things, maybe trying the virus maybe without the treatment, and then with the CNO to see if it's possible to alter the inhibition without altering anything else, and have it be safe for patients.
Amazing. Well, thank you so much. It's very, very exciting work, and we look forward to watching it move closer to the translational spectrum.
Thank you so much for having me again.
No problem. Thanks for joining us, and have a great rest of your day.